I'll introduce myself again. My name is Chiteri. I am in Dr. Ati Singh lab, and I come from Kenya. And I'm here because I have an interest in understanding breeding and technologies that, I can export back to my people. And the question I asked earlier was, what is the average size of an American farmer, the small-scale farmer in America? Pop it up. An idea? The smallest farm size in America, apart from gardens. Okay, I think I'll give you this. So it's about 40 acres, right?
Small farmer, you probably looked at
300 to 400 acres. So where I come from, a small-scale farmer from about 1 to about 10 acres, which means here is, lots of times higher. But where I come from is smaller, and so I'm interested in understanding some of these technologies and how we can, adapt to those small farms to ensure maximum sustainability and profitability from those small farms. But here today, my interest is in talking about genomics in breeding. So when I throw out the word genomics, what comes to your minds? GMOs, yeah? Someone else? We actually have that here today. We'll talk about it. So if we want to relate something here, you are the genetics students, agronomy students, right?
They are all
agronomy students. You probably come across the word genetics class, right? So you're talking about genes, right? And so when you have all the genes in an organism, be it human or plant, that makeup is what is called a genome. And so when you take the genome and put on information technology to analyze the genome, then you get what? Genomics. That's what the omics comes in. So that genomics is just the study of the whole genome of an organism, but to our case today, we'll be focused more on plants. And so first of all, a more interesting thing is the cell. We all are familiar with what the cell is, and inside the cell we find what? The chromosomes. Now each chromosome is made up of a molecule that's very important for us, and that's the DNA, right? With all the bases that are on it. Those bases that make up the DNA, we have specific size of them that end up making what we call a gene. Now the gene is what would relate to what all other my colleagues have said, because when you go out there as a student or as a farmer, you are interested in what you see, right? Be it the quality that you want, the height, is what you're seeing. But it is somehow controlled. It is actual control from the gene, from the genetics. And so that is what is more important for us at this particular point. We want to understand, how does the genetics affect what we see, what we desire, what we want to put on the table for dinner, yeah? And so I break this down today into 3 items. And the first one will be about sequencing, bioinformatics and molecular markers, gene engineering, and CRISPR. And so I'll just start right here. Now that we know DNA, we can go in DNA, and we know DNA is made up of what? An idea?
Repeat the question, please.
What is DNA made up of? DNA. I'm assuming we know what DNA is, right? So do you have an idea of what it's made up of? It's basically made up of a sequence of, go ahead. Okay. It's basically made up of a sequence of bases, right? Bases, an idea of what those bases are? So those bases are, we have a cytosine, guanine. What are the others? Yeah? Yeah, there we go. So they have those bases. So when we can, for example, go ahead and know how are these bases arranged, that's what we call is a sequence. So using the current technology, you can be able to go in and look at the sequence of the DNA, and that gives us the term what's called sequencing. Now once we have all that sequence, we have to analyze that sequence, and that's where the term bioinformatics comes in, You have all these massive data of the DNA from a cell, how it is organized, and now you start relating it to some of the traits that all other my colleagues are assembling. And in some cases, we have gone to extent where we know exactly what gene affects what phenotype, what trait, what characteristics that we want. In other cases, we don't know. So in the cases we don't know, we can find things that we are calling molecular markers, will be pieces of DNA or the sequence that are close enough to that gene so that they are always inherited together, or that whenever you see that trait, then you are sure, whenever you see these markers, then you are sure it is associated with this gene or these characteristics. And that's where the term molecular markers comes in. Now, I brought this up first because earlier on, breeders, before Dr. Singh, used to walk fields. Even now, they still do. And they go looking for plants because of phenotypes that they're interested in. Now, these phenotypes or characteristics are accumulated over time through things that we are called mutations, right? So they didn't do anything. They basically walked the fields, looked for characteristics, and each plant has different characteristics. And so what you do, you cross the plants to get the F1s, and you keep going looking for the phenotypes that you want in the different plants. And so that allowed me to turn around this. So that's, it's okay. Let's see if we can. So that brings us to what I was saying earlier on, that earlier on we used to do breeding just via mutations, that they are naturally occurring, even in human beings, that there are mutations that occur, and that's why they keep changing phenotypes. And so, but nowadays you can even induce the mutations, right? If you get, you X-ray the plants, expose the plants to X-rays, then they change the genetic makeups. And that, indeed affects the different phenotypes. But now we have the mutation, the molecular markers that you can use to breed for different things that you want. But as we go on, things have advanced, right? We need more people we need to feed. We are choosing the kind of foods that we want on the table. And that means that we have also to accelerate the way we breed the different things that we want. And so that's where the term genetic engineering comes in, or GMOs. In full, what is it? What is the GMO in full? Go ahead. Anyone? Yeah, genetically modified organisms, which means that we have been able to go to the basic unit, which is the DNA, and change it somehow the way we want it. Now, can we name some crops that we know around that are genetically modified? Some crops that you know that are genetically modified around? Corn, soybeans? Yeah, that's right. And do you know for what traits? Go ahead. Yes, yes. So basically we have crops, and we try and breed crops that are to the advantage that we want. For example, you have a bunch of insects that chew up your corn. So if we can be able to plant a corn that even in the presence of those insects, they are not affected, that's what we're having here. Almost 90% of corn and soybean in the US is genetically modified. So to that extent, that means that we are breeding in traits that we want. And we can be able to cross, we can be able to get these genes from different organisms to our organisms of interest, and that's where genetically modification comes in. But when we are doing this, it's a tossing a coin. It can go either way, right? That's so you get to introduce these genes and they can go anywhere and do all that thing. But now we have another cooler technology which is called CRISPR. CRISPR-Cas CRISPR-Cas allows us to, the way this image shows here is that allows us to go inside and behave like surgeons. We perform surgery on the DNA of the plant with the precision of a surgeon, that we know exactly if we are inserting something else, we want to know where it is, what it's doing, and everything about it. And so that's what the new technology that I think, like Dr. Singh said, is going to revolutionize how we do breeding. Because we're going to go in, what do you like? Say you like red flowers, right? You like a plant that has red flowers, and you know another plant that you don't like but has red flowers, and if you can get that gene into it so that, you know, on your balcony you have red flowers, then you are all happy. So you can basically do that right now. But breeders are going to enhance more of that as we go forward, because then we can get plants that, you know, are climatic resilient, the way things are changing right now. So I think I'm going to wind up with what I'm saying we're going to take home today. My note here is one is that due to the increase in population and all other things, as breeders we have to accelerate the way we breed crops. It used to take about, you know, 10, 20 years to breed a single variety or a single line out into the field, but right now we are talking about 5, 7, 10 years to release a variety, which means that, you know, we keep changing the way things keep changing too. 3, we are saying that the introduction of genetic engineering, CRISPR-Cas, and all these things will also help us in achieving our goal. And lastly is that if we keep using genomes and genomics, is a big step towards achieving our goal currently. So if you have any other question, please throw it at me, and feel free to pass by here before we leave.